Good day, everyone. So, for today's discussion is all about the nervous system. In a complex nervous system, millions of neurons work together to produce effective responses to stimuli in the external environment. The nervous system also regulates heart rate, breathing, and hundreds of other internal activities. Researchers have focused extensively on just how neurons interact, and we're only beginning to understand the mechanisms that permit the many functions of the nervous system. In this chapter, we compare various animal nervous systems. We then examine the structure and function of the vertebrate nervous system, with emphasis on the function of the human brain. We explore some of the frontiers of neurobiology, such as the mechanisms involved in information processing. So first, we have the invertebrate nervous system. An animal's lifestyle is closely related to the organization and complexity of its nervous system. Hydra and other cnidarians have a nerve net consisting of interconnected neurons with no central control organ. An advantage of the nerve net is that the cnidarian respond effectively to predator or prey approaching from any direction. So in some jellyfish, a slow transmission of nerve net coordinates movement of the tentacles, and a second nerve net, which is faster, coordinates swimming. So the radial nervous system of the sea star and other echinoderms is a modified nerve net, and the system shows some degree of selective organization of neurons into more than a diffuse network. It consists of a nerve ring around the mouth, and from which a large radial nerve extends into each arm. Bilaterally symmetrical animals have a bilateral nervous system. In ternarian flatworms, the head region contains concentrations of nerve cells called cerebral ganglia, and these serve as a primitive brain and have some control over the rest of the nervous system. So typically, two solid ventral longitudinal nerve cords can extend from the ganglia to the posterior end of the body, and transverse nerves connect to the two nerve cords and connect the brain with the eye spots. This arrangement is called a ladder-type nervous system. So next is the annelids and arthropods typically have a solid ventral nerve cord, and the cell bodies of many of the neurons are massed into ganglia. Afferent and efferent neurons lie in lateral nerves that link the ganglia with muscles and other body structures, and if our earthworm's brain is removed, the animal can move almost as well as before. However, when it bumps an obstacle, it persists in a futile effort to move forward rather than turns aside to respond adaptively to environmental change. The earthworm needs its brain, of course. So we also have the cephalopod mollusk. The squids are octopods and that has well-developed nervous system that include well-developed sense organs. So neurons concentrated in ganglia are massed in a ring surrounded the esophagus, and they make up a brain that contains about 168 million nerve cells. Like the other vertebrate brain, the cephalopod brain has lobes and intricate folds, and particular areas are specialized as centers for specific functions. The octopus is capable of considerable learning and can beat out difficult tasks. In fact, cephalopods are considered the most intelligent invertebrates. Now let's move on to the vertebrate nervous system. So the vertebrate nervous system has two main divisions, the central nervous system and the peripheral nervous system. The CNS consists of a highly developed brain that is continuous with the dorsal tubular spinal cord, serving as central control. These organs integrate incoming information and determine appropriate responses. So the PNS is made of the sensory receptors, for example, tactile, auditory, and visual receptors, and the nerves, which are the communication lines. Various parts of the body are linked to the brain by cranial nerves and to the spinal cords by spinal nerves. Afferent neurons in these nerves continuously inform the CNS of changing conditions. Then efferent neurons transmit the decisions of the CNS to appropriate muscles and glands, which make the adjustment needed to maintain homeostasis. PNS is subdivided into somatic and autonomic divisions. So most of the receptors and nerves concerned with changes in the external environment are somatic, meanwhile those that regulate the internal environment are autonomic. Both divisions have afferent nerves which transmit messages from receptors to the CNS, and efferent nerves which transmit information back from the CNS to the structures that respond. The autonomic division has two kinds of efferent pathways, the sympathetic and parasympathetic nerves, will be discussed later in the chapter. So let us also consider the evolution of the vertebrate brain. In the early vertebrate embryo, the brain and spinal cord differentiate from a single tube of tissue, the neural tube, and anteriorly, the tube expands and develops into the brain, and posteriorly, the tube becomes the spinal cord. Brain and spinal cord remains continuous and their cavities communicate. As the brain begins to differentiate, three bulges become visible. These are the hindbrain, the midbrain, and the forebrain. So the hindbrain develops into the medulla, pons, and the cerebellum. As you can see on the table, the hindbrain subdivided to form the metacephalon, which gives rise to the cerebellum and pons, and the melencephalon, which gives rise to the medulla. The medulla, pons, and midbrain make up the brain stem, the elongated portion of the brain that looks like a stalk holding up the cerebrum. And the medulla, the most posterior part of the brain, is continuous with the spinal cord, and its cavity, the fourth ventricle, is continuous with the central canal of the spinal cord and with a channel that runs through the midbrain. On the other hand, the cerebellum coordinates muscle activity and is responsible for muscle tone, posture, and equilibrium. So a bird without a cerebellum cannot fly, and its wings thrash about jerkily. So when the human cerebellum is injured by a blow or by disease, muscular movements are uncoordinated. So any activity requiring delicate coordination, such as lifting a needle, becomes very difficult if not impossible. The midbrain or mesencephalon is the largest part of the brain in fishes and amphibians. It is the main association area, linking sensory input and motor output. In reptiles, birds, and mammals, the midbrain serves as a center for visual and auditory reflexes. So the medulla, pons, and midbrain make up the brainstem. The forebrain differentiates to form the diencephalon and telencephalon. So diencephalon develops into the thalamus and hypothalamus. The thalamus is a relay center for motor and sensory information. The hypothalamus controls autonomic functions, which links the nervous and endocrine system, controls temperature and appetite, and fluid balance, and is also involved in some emotional and sexual responses. The telencephalon develops into the cerebrum and olfactory bulbs. In most vertebrates, the cerebrum is divided into right and left hemispheres. And in fishes and amphibians, the cerebrum mainly integrates incoming sensory information. In birds, the corpus stratum controls complex behavior patterns such as flying and singing. So now is the human central nervous system. Our soft, fragile human brain and spinal cord are well protected. Encased within bone, they are covered by three layers of connective tissue, the meninges. The three meningeal layers are the tough outer dura mater, the middle arachnoid, and the thin vascular pia mater, which adheres closely to the tissue of the brain and spinal cord. The space between the arachnoid and the pia mater is the subarachnoid space, which contains cerebrospinal fluid. CSF is a shock-absorbing fluid that cushions the brain and spinal cord against mechanical injury, and it also serves as a medium for exchange of nutrients and waste products between the blood and brain. CSF is produced by special networks of capillaries, collectively called the choroid plexus, and that extend from the pia mater into the ventricles. After circulating through the ventricles, the CSF passes into the subarachnoid space, and it is then reabsorbed into large blood sinuses within the dura mater. So the spinal cord transmits impulses to and from the brain. The tubular spinal cord extends from the base of the brain to the level of the second lumbar vertebra. A cross-section view through the spinal cord reveals a small central canal surrounded by an area of gray matter, shaped a bit like the letter H. The gray matter consists of the large mass of cell bodies, dendrites, amylated axons, and glial cells. The white matter, on the other hand, found outside the gray matter, consists of myelinated axons arranged in bundles called tracts or pathways. The ascending tracts conduct impulses up to the cord to the brain. For example, is that the spinothalamic tracts in the anterior and lateral columns of the white matter conducts pain and temperature information from the sensory neurons in the skin. So next is the pyramidal tracts, which are the descending tracts that convey impulses from the cerebrum to motor nerves at various levels in the cord. We describe the spinal nerves later in this chapter. So in addition to transmitting to and from the brain, the spinal cord controls many reflex activities. So a reflex action is a relatively simple involuntary motor response to a stimulus. Although most reflex actions are more complex, let us consider a withdrawal reflex, in which a neural circuit consisting of three types of neurons carries out a response to a stimulus. So suppose that your hand accidentally touches a flame, almost instantly, and even before you became consciously aware of what has happened, you jerk your hand away. In this brief instant, a sensory neuron transmits a message from the pain receptors in the skin to the spinal cord. Within the spinal cord, a sensory neuron transmits the signal to an interneuron. So the interneuron integrates the information and signals an appropriate motor neuron, which conducts the information to groups of muscles, and the muscles respond by contracting and moving the limb toward the midline of the body, and that is moving the hand away from the from the flame. So actually, many neurons in sensory association and motor nerves participate in such a reflex action. So generally, we are not even consciously aware that all these responding muscles exist. In summary, signals are transmitted through a visual reflex in the following sequence. So as you can see, at the same time, the reflex pathway is activated. A message is sent up the spinal cord to the conscious area of the brain as you pull your hand away from the flame, so you become aware of what has happened and feel the pain. This awareness, however, is not part of the reflex response. The spinal cord has some plasticity and is capable of training. So, for example, sensory feedback from exercise may increase the strength of neural connections in the spinal cord. If descending connections with the brain are intact, some victims of spinal cord injury could potentially develop at least some limited ability to walk again.
As a continuation to Eloisa's discussion, I will talk about information processing and the peripheral nervous system. So recently, neurobiologists are beginning to understand how the nervous system functions, such as how neurons signal one another, connect, and carry out basic functions such as regulating heart rate, respiration, and the sleep-wake cycles, as well as how we learn and remember. An interesting question has been, to what extent are these functions hardwired or preset? Even animals with very simple nervous systems can learn. They learn to repeat behaviors associated with reward and to avoid behaviors that cause pain. Such changes in behavior are possible because of neural plasticity. Now, what is neural plasticity? It is the ability of the nervous system to change in response to experience. Such change modifies structure and function, so the more experience you get, the more complicated your neurons become. Many areas of the brain that are once thought to be hardwired are now known to be flexible and are capable of change. So the following are common examples of neural plasticity in human motor skills, and it includes learning to walk, riding a bicycle, or catching a baseball. At first, we were probably clumsy, but with practice, our performance becomes smoother and more precise. For that to happen, changes must occur in the brain. In a similar manner, the ability to learn languages, solve problems, and perform scientific experiments also depends on neural plasticity. Now, what are the mechanisms by which neurons signal one another to learn diverse activities? Well, there is a Canadian psychologist, Donald O. Hebb, who proposed in 1949 that when two neurons connect by a synapse, that is, presynaptic and postsynaptic neurons, are active simultaneously, the synapse is strengthened. Then in 1973, British researchers T.V.P. Bliss and T.J. Lomo found experimental evidence for Hebb's hypothesis. They did it by investigating rabbit brains, and these researchers applied a series of high-frequency electrical stimuli to neurons in the hippocampus, which is a region in the brain that converts information into memories, and the result that they found was that there is a long-term increase in synaptic strength. Because neurons signal one another at synapses, these junctions between neurons are currently the focus of intense research. Neural plasticity may in fact be synaptic plasticity because changes that occur during learning and remembering appear to take place at synapses. It has also been found that as the brain matures, it loses some of its plasticity. Here, let us examine the relationship between learning and the nervous system. Firstly, what is learning? Learning is the process by which we acquire knowledge or skills as a result of experience. For learning to occur, we must be able to remember what we experience. Memory, on the other hand, is the process of encoding, storing, and retrieving information or learned skills. There are two types of memory. The implicit memory, also known as the procedural memory. It is the unconscious memory for perceptual and motor skills, such as riding a bicycle. So if you notice when you ride a bicycle, we don't consciously think of how to ride it. We just know, and it's natural for us. On the flip side, implicit memory is about how to do something, and it involves repeating the behavior until it becomes a routine. Explicit memory, also called declarative memory, involves factual knowledge of people, places, or objects, and it requires conscious recall of the information. There is a part of the brain that is critical in forming and retrieving explicit memories. It is called the hippocampus, and we will delve deeper into it more in the following slides. But before that, let us first learn about information processing. So, information processing involves short and long-term memory. At any moment, we are bombarded with thousands of bits of sensory information. For example, at this very moment, your eyes are receiving information about the words that you see on the slide right now, or the objects around you, and the intensity of the light in the room where you are right now. At the same time, you may be hearing a variety of sounds, just music, or your friends talking to you, and many more. Also, your olfactory epithelium may sense cologne or the smell of coffee or whatever. Or maybe you're eating as you watch this discussion, and sensory receptors in your hands may be receiving information regarding the weight and position of the object that you are holding right now. Most of these sensory stimuli are not important to remember and so are filtered out. When we select information to remember, we must process it. When we begin to process information, we encode it. We recognize patterns and meaningfully associate the stimuli to past experiences or knowledge. Information processing involves both short-term memory, which lasts only for minutes, and long-term memory, which may last for many years. Typically, we can hold only about 7 chunks of information at a time in short-term memory. Short-term memory let us recall information for a few minutes. For example, when we look up a phone number, we usually remember it only long enough to dial. If we need the same number in an hour later, most of us have to look it up again. Now, I already introduced this earlier and we will learn further about this crucial part of the brain, the hippocampus. The hippocampus temporarily holds new information and may integrate various aspects of an experience, including odors, sounds, and other information. Memory consolidation, which depends on the hippocampus, allows memories to be stored for long periods. Consolidations involve expression of genes and protein synthesis and depends on new neural connections at synapses. Time is required for the brain to consolidate a new memory. Here, let's talk about a condition that is associated with memory. So, if a person suffers a brain concussion, memory of what happened immediately before the incident may be completely lost. This is known as retrograde amnesia. When the hippocampus is damaged, short-term memory may be unimpaired and the patient can still recall information stored in the past. However, new short-term memories can no longer be converted to long-term memories. Now, there's a very interesting question that I want to ask you. Where are memories stored? Well, actually, no specific area in the brain can be labeled as the memory bank. Rather, memories seem to be stored within many areas of the brain. For example, visual memories may be stored in the visual centers of the occipital lobes, and auditory memories may be stored in the temporal lobes. Researchers think that memories are integrated in many areas of the brain. The following are some other locations that memories may be stored. It includes the amygdala and the hippocampus, association areas of the cerebral cortex, and the thalamus and hypothalamus. Wernicke's area in the temporal lobe has been identified as a very important association area for complex thought processes. Neurons within the association areas form interconnected pathways that permit complicated reverberation. There are neurobiological changes that occur during learning. It is facilitated by the repeated electrical stimulation of neurons causing functional change at synapses. When a presynaptic neuron continues to transmit action potentials at a high rate for a minute or longer, there is a long-lasting strengthening of the connection between the presynaptic and postsynaptic neurons. This increased strength of the synaptic connections is known as long-term potentiation. Potentiation means to strengthen or make more potent. In contrast, low-frequency stimulations of neurons result in a long-lasting decrease in the strength of their synaptic connections, and it is called long-term depression. Information storage and forgetting depend on the strengthening and weakening of synaptic connections brought about by LTP and LTD. Furthermore, induction of LTP and LTD requires activation of 2 types of receptors, the NMDA and the AMPA receptors. These receptors present in the plasma membranes of postsynaptic neurons control the passage of calcium ions into neurons. NMDA receptors respond to the neurotransmitter glutamate by opening calcium ion channels. However, when the postsynaptic neuron is at its resting potential, NMDA ion channels are blocked by magnesium ion. Here, what you see is a model for the mechanism of LTP. A presynaptic neuron releases glutamate, which binds with AMPA receptors. The postsynaptic neuron becomes depolarized. If the polarization of the postsynaptic neuron is sufficiently strong, magnesium ion move away from the NMDA receptors and unblocking them. Glutamate can then bind with these receptors, opening calcium ion channels and letting calcium ions move into the cell. And we can see here that the calcium ions appear to be a very important trigger for LTP. Further, calcium ions act as a second messengers that initiate long-term changes ultimately responsible for LTP. For example, calcium ions activate a calcium ion-dependent second messenger pathway that results in the insertion of more AMPA receptors in the postsynaptic membrane. This is important because additional AMPA receptors increase sensitivity to glutamate. More EPSPs are produced, which strengthens the synapse, which helps maintain the LTP. And aside from that, calcium ions also activate a pathway that leads to release of a retrograde signal, or a signal that moves backward from the postsynaptic neuron to the presynaptic neuron. The soluble gas nitric oxide has been identified as a retrograde signal. This signal enhances neurotransmitter release by the presynaptic neuron. Now let's move to the next subtopic, which is the peripheral nervous system. The peripheral nervous system consists of the sensory receptors, the nerves that link these receptors with the central nervous system, and the nerves that link the central nervous system with effectors, such as muscles and glands. There are two divisions of the peripheral nervous system: the somatic division, which helps the body respond to changes in the external environment, and the autonomic division, which is when the nerves and receptors maintain homeostasis despite internal changes. Let's look further on each divisions in the following slides. So first, let us look into the somatic division. The somatic division helps the body adjust to the external environment. The somatic division of the PNS includes the receptors that react to changes in the external environment, the sensory neurons that inform the CNS of those changes, and the motor neurons that adjust the positions of the skeletal muscles that help maintain the body's posture and balance. What you see here is the cranial nerve. So in mammals, there are 12 pairs of cranial nerves that emerge from the brain. You can see each of these in the illustration. These cranial nerves transmit information regarding the senses of smell, sight, hearing, and taste from the special sensory receptors, as well as information from the general sensory receptors, especially in the head region. For example, cranial nerve 2, or the optic nerve, transmits signals from the retina of the eye to the brain. Now, what you're looking at is the illustration of the spinal nerves. In humans, there are 31 pairs of spinal nerves that emerge from the spinal cord. They are named after the general region of the vertebral column from which they originate. They comprise 8 pairs of cervical, 12 pairs of thoracic, 5 pairs of lumbar, 5 pairs of sacral, and 1 pair of coccygeal spinal nerves. The ventral branches of several spinal nerves from tangled networks is called plexi. Within a plexus, the fibers of a spinal nerve may separate and then regroup with fibers that originate in the other spinal nerves. Thus, nerves emerging from a plexus consists of neurons from different spinal nerves. Moving on to the next division, the autonomic division, which regulates the internal environment. The autonomic division helps maintain homeostasis in the internal environment. For example, it regulates the heart rate and helps maintain a constant body temperature. The autonomic system works automatically and without voluntary input. Its effectors include the smooth muscles, the cardiac muscles, and glands. Like the somatic system, it is functionally organized into reflex pathways. Receptors within the visceral relay information via different nerves to the central nervous system. The information is integrated at various levels. Then the decision is transmitted along efferent nerves to the appropriate muscles or glands. Afferent and efferent neurons of the autonomic division lie within cranial or spinal nerves. For example, afferent fibers of cranial nerve X, which is the vagus nerve, transmit signals from many organs of the chest and upper abdomen to the central nervous system. Its efferent fibers transmit signals from the brain to the heart, stomach, small intestine, and several other organs. The efferent portion of the autonomic division is subdivided into the sympathetic and the parasympathetic systems. Many organs are innervated by both types of nerves. In general, sympathetic and parasympathetic systems have opposite effects. For example, the heart rate is speeded up by messages from sympathetic nerve fibers and slowed by impulses from its parasympathetic nerve fibers. In many cases, sympathetic nerves operate to stimulate organs and to mobilize energy, especially in response to stress, whereas the parasympathetic nerves influence organs to conserve and restore energy, particularly during quiet, calm activities. Instead of using a single efferent neuron as in the somatic system, the autonomic system uses a relay of 2 neurons between the CNS and the effector. The first neuron, called the preganglionic neuron, has a cell body and dendrites within the central nervous system. Its axon, which is part of a peripheral nerve, ends by synapsing with a postganglionic neuron. The dendrites and cell body of the postganglionic neurons are in a ganglionic outside of the CNS. Its axon terminates near or on the effector. The sympathetic ganglia are paired in a chain of them. The paravertebral sympathetic ganglion chains runs on each side of the spinal cord from the neck to the abdomen. Some sympathetic preganglionic neurons do not end in these ganglia, but pass on to collateral ganglia in the abdomen, which is close to the aorta and its major branches. Parasympathetic preganglionic neurons synapse with postganglionic neurons in terminal ganglia near or within the walls of the organs they innervate. The sympathetic and parasympathetic systems also differ in the neurotransmitters that they release at the synapse with the effector. Both preganglionic and postganglionic parasympathetic neurons secrete acetylcholine, whereas sympathetic postganglionic neurons release norepinephrine, although preganglionic sympathetic neurons secrete acetylcholine.